Today we're going to talk about the various mechanisms for genetic exchange in bacteria. These are transformation, conjugation, and transduction. Okay, transformation is first. It's the simplest, easiest to understand, I think, where DNA is acquired from the environment. It's not encapsulated in a phage particle, and it's not acquired from another cell directly. It's actually DNA that's floating around in solution or suspension adjacent to the cell. So the DNA could be chromosomal DNA or plasmid DNA, but if it's chromosomal DNA, it will need to recombine into the chromosome in order to become part of the genetic material of the cell. If it's a plasmid, however, it just needs to enter the cell because it's able to replicate on its own. So the familiar type of plasmids are cloning vectors, and we know that they carry drug resistance for experimental purposes, and that they can be made to be taken up by E. coli cells by treating the cells in such a way that they're chemically competent. Competent meaning are able to take up exogenous DNA. Okay, so it's for this purpose very important that they can replicate independently of the host chromosome, meaning that they do not have to recombine with the host chromosome in order to become residents of that particular cell. Now, for experimental purposes, the usual lab plasmids carry antibiotic resistance, for example, ampicillin resistance, so that we can then select for those rare cells that have actually acquired the plasmid from the outside world. So here's our plasmid vector. We've seen this before. It's called PUC18. It contains the beta-lactamase gene, which encodes it encodes a protein beta-lactamase that is able to open the beta-lactam ring and render the penicillin or ampicillin inactive. But importantly, have a replication origin, which allows this molecule to survive and replicate inside cells without integrating into the host chromosome. So here's a reaction carried out by the beta-lactamase one more time. The penicillin-type antibiotics, including ampicillin, have a beta-lactam ring, which is important in its function in binding cell wall proteins. And the beta-lactamase opens that up, essentially rendering the penicillin ineffective in causing the cell walls to weaken. So this plasmid confers a new phenotype on cells, which is to say it lets the cells become resistant to the ampicillin, whereas the wild-type starting cells were sensitive. So we can detect this sort of event simply by plating the cells, on the appropriate kind of media that's going to select for the ampicillin or any other drug resistance that might be present on our plasmid. So our competent cells will be mixed with our plasmid DNA and treated by heating and cooling and whatnot, but then importantly put onto the selective media, which contains the drug ampicillin. Okay, so what happens is we observe colonies, and these colonies are called transformants, and we know that they contain the plasmid DNA because they grew up resistant to the ampicillin. Most of the cells are killed in this situation because transformation is a rare event in E. coli, and the rare cell that does take up the plasmid expresses the beta-lactamase gene and is now able to grow in the presence of the drug because the cells are now protected from the drug because of this enzyme produced by the plasmid. So the ampicillin selects for transformants. Non-transformed cells are killed, and the transformed ones live and form colonies. Here's a silly picture. This is the rare cell that actually got the little circle plasmids are making the little protein dots are the beta-lactamase, which go to the cell surface, and those cells are able to survive the drug because they are protected from it by this enzyme. The other cells, which did not get the plasmid, are shown with red weakening cell walls, causing them to eventually break open and die. So we are selecting for the presence of the plasmid by means of its phenotype and changing the phenotype of the cell from ampicillin sensitive to ampicillin resistant. The plasmid is taken up by an E. coli cell. The mechanism for genetic alteration of the phenotype is called transformation, transduction, conjugation, transposition, or complementation. Now we're going to get this one right. Of course, it's transformation. Keep remembering that. In typical plasmid cloning vectors, the role of the ampicillin resistance gene is to permit survival of cells containing the plasmid, kill the surrounding untransformed cells by producing ampicillin, guarantee that the plasmid can replicate, produce a location for cloning the mammalian gene, select specifically for clones containing the mammalian gene. Okay, so the answer is A, permit survival of the cells containing the plasmid. The role of the origin of the plasmid vector is to provide a location where replication can start, provide a promoter for the AMP gene, provide a location where the insert can be added, provide the selectable marker. Yes, provide a location where replication can start. Okay, transformation is a little bit different when it's not a plasmid that's going in. We're asking the cell to take up genomic or chromosomal DNA from the outside world and then have it become part of the genome of the recipient cell. So the same process can cause any gene to go into the cell, but it needs to be recombined into the chromosome in order to become a permanent part of that host cell, unlike plasmids which can replicate on their own. Now this is, of course, again, a very rare event, and we need to be able to select for it in the sense of setting up a situation where those rare cases where the DNA has been taken up will give rise to a colony that we can recognize as being distinct from all the rest of them. So this needs to take into account the genotype of the host cell as well as that of the incoming DNA to set up a situation where such an event can be recognized and selected for. Okay, what's represented here is a sort of schematic of what's happening in a transformation experiment in which we're transforming a recipient cell with chromosomal DNA from a different source. So the donor cell, we're going to assume is going to be wild type A plus B plus C plus D plus E plus, etc. And that the DNA from that cell has been sheared into chunks so that any particular chunk contains a certain number of genes, but by no means all of them. Okay, so we need to set up a selection. We have here a recipient cell, which is deficient for A, B, C, D, and E. So that particular cell might be auxotrophic for multiple things, or it might have other genetic markers that we can then easily identify. So what's going to happen is that the DNA fragments from donor cells will go inside the recipient, more or less at random. The recipient cell takes up the donor DNA. But there it sits because that incoming donor DNA, which is A plus B plus, can't replicate. It needs to exchange itself, essentially, with what's in the chromosome already in order to become permanently part of the host cell chromosome. So what happens is some part of that, all of it or part of it, will recombine with the host chromosome. In this case, we're showing B, the B plus, to have undergone a recombination event in such a way that the donor DNA replaces what was there originally, which was the B minus. So the DNA that was in the chromosome is essentially destroyed and replaced by this wild type allele that's coming from the donor DNA. Now this is the type of event that we can easily select for, assuming auxotrophic markers are involved. Okay, so for example, we want to get B plus transformants. Recipient is little a, little b, little c, little d, little e. Let's say those are his minus Lu minus pro minus R's minus your minus. So B is going to be Lu. So the incoming DNA has wild-type alleles of all those genes, including the Lu plus, which is the one we're going to be selecting for. So the medium can be set up to select for that particular rare event, in which we will prevent the non-transformed cells from growing, but allow the transformed cells that have the Lu plus cells to grow. So we certainly have to have a medium that does not contain leucine, because we want to select for those cells which have acquired the wild-type allele from the outside world. Meanwhile, the cell is auxotrophic for a whole bunch of things, so we need to add to the medium histidine, proline, arginine, and uracil to supplement the auxotrophies other than Lu. So in order to do this, we will have a minimal medium with glucose that will have histidine, proline, arginine, and uracil, but leave out Lu. And that way we're going to be selecting for the transformation of the B, which is to say Lu, to be selected. Okay, an Rh minus Lu minus Pro minus recipient was transformed to a Pro plus phenotype. The medium used was therefore rich medium containing glucose, minimal containing glucose and proline, minimal containing glucose, proline, arginine, and leucine, minimal containing glucose, arginine, and leucine, rich medium containing glucose, arginine, and leucine. Okay, so the answer is D, minimal medium containing glucose, arginine, and leucine. Yes, because we're selecting Pro plus. Get confused for a sec there. Conjugation is the second type of genetic transfer in bacteria that we will consider, and this involves gene transfer by cell-to-cell contact. This is not DNA floating around the medium. We have two cells that actually have to come in contact with DNA, with each other and transfer DNA from one cell to the other. Now, the important genes here are those located on the F factor for fertility, and as well as the related conjugatable factors called R factors. Reminding this is what the F factor looks like. It's a very large plasmid and includes this enormous section of DNA which encodes proteins that are essential or necessary and sufficient, shall we say, for transfer of the entire plasmid to a different cell. Okay, in terms of nomenclature, cells containing the F plasmid are called F+, meaning they have it, and cells without it are called F-. One of the things that the F plasmid encodes in the Tra region are pili, which are cell surface protrusions which contact other cells. So proteins that are actually encoded by the F factor will catalyze transfer of the F to new cells, converting them to F+. So the cell that's F+ transfers the F to a second cell, does not lose it itself, but converts the F- cell then to a F+. So this is, you can see why that would spread through populations of bacteria. Because once the F- becomes F+, it now can also transfer F to new cells. And this will carry whatever happens to be passenger on that F molecule as well. A schematic picture of this, we have a cell containing the F plasmid. It creates cell surface pili, most of which are not shown here, which actually contact F- cells and allow an opening between the two cells where the F plasmid can then replicate. So what it does, it sends one strand or one copy of itself to the recipient, and this strand is being copied the other strand is being copied as it goes along. So one strand is being transferred and copied as it goes, and the remaining F inside the cell is itself being copied. So this is obviously a much more complicated process than we're showing here. It's very okay. But the result is, of course, that the F plasmid has replicated, transferred one copy of itself to the other cell. So now the F- cell is converted to F+, which can, of course, then transfer the F to further cells in the population. Okay. There's also another interesting feature of the F plasmid, and that is its ability to actually insert itself into the chromosome of cells in which it lives. So the F factor normally replicates on its own independently of the host cell chromosome, but it can actually integrate by means of recombination between IS elements located on the F and the bacterial chromosome. So this recombination event that's being depicted basically results in that you have a recombination between IS elements, and with the result that the F is now part of the chromosome. Now this is called a high-frequency recombination strain because it's actually able to transfer bacterial genes, not just itself, into the recipient cell. This slide represents what happens when you have an integrated F factor called an HFR, which is then going to transfer itself, part of itself anyway, from the F+ cell to an F-. So what happens is the HFR cell, because it contains the entire F, albeit integrated in the chromosome, will make its pili, will recognize F- cells, and will initiate transfer of itself into the F- cell. But the way it works is there's a specific location in the F where the transfer originates, and that's somewhere in the middle of the F. So transfer will start at that location and then start working its way around the bacterial chromosome, starting within the F, but soon entering bacterial DNA and starting to transfer bacterial DNA instead. Now, the mating pairs generally don't stay together for more than 15 or 20 minutes or so that only a certain amount of the bacterial chromosome can be transferred in any one particular cross, generally speaking. So the implication of that is that what you've transferred to the F minus cell is a bit of the F factor starting in the middle, and a portion of the bacterial DNA, but by no means all. So as a result of this, the last part of F is left behind. So what goes into the recipient cell includes part of F, but not the entire F. Now that part of the DNA that's been transferred has the opportunity to recombine with the recipient chromosome and transfer its alleles to that chromosome, just as if we were transferring donor DNA from a transformation. But the cell does not become F plus because the amount of the F is incomplete. Only part of itself got transferred, and the rest was left behind. So what you can do is transfer a good chunk of bacterial genes actually, but not all usually. But you do not hardly ever transfer the entire F. So the F minus cell can acquire genes from the HFR cell, but very rarely becomes F plus in the process. On the other hand, the alleles that were present in the donor strain can be found recombined into the recipient chromosome. Okay, so it must be recombined because otherwise it won't replicate, and it does replace resident alleles in the process. Now, it takes 100 minutes. Remember, the E. coli genetic map is 100 minutes long, so it takes officially 100 minutes for transfer to be complete. But this rarely happens in practice. Usually the recipients don't get the whole chromosome, they only get part of it. They usually remain F minus because part is left behind. Okay, which of the following mechanisms of gene transfer involves cell-to-cell contact? Transformation, conjugation, transposition, recombination, transduction. Yes, conjugation. The DNA of the F plasmid contains all of the following except an origin or replication, genes encoding cell surface villi proteins, genes for amino acid biosynthetic proteins, genes for transfer of DNA from one cell to another, sites for RNA polymerase to bind. Yes, of course the answer is C. Remember, the sites for RNA polymerase to bind just means promoters for those genes that are important. So any DNA, plasmid, or whatever is going to have promoters to allow itself to express its RNA and its proteins. So the answer is genes for amino acid biosynthetic proteins. Our next topic for today is phage and transduction. Okay, so transduction means the bacterial DNA can be moved from one cell to another by means of a phage particle. Now, there are two types of transduction, generalized, involving phages such as P1, which we'll talk about, meaning that any piece of bacterial DNA can be transferred. There's also specialized transduction, in which only certain genes can be transferred. And these mechanisms are distinguished by the mechanisms whereby they actually occur. Okay, remember viruses, bacteriophages. They rely on the cell for most aspects of certainly protein synthesis, and they replicate independently of the cell chromosome using their own specific origin, whereas some can integrate into the host chromosome. Lambda and P1 are the most famous E. coli examples, plus a few more. Okay, here's our picture we saw last time. This is a lambda phage, but it's a member of a class of phages which have very similar morphologies, including a head, collar, tail, and these base plate and tail fibers, which allow this phage to recognize a sensitive cell and inject the DNA, which is inside it, into the cell in order to produce more phage, usually killing the new host cell. Now we're going to describe a sort of general scheme of infection of a susceptible cell by what's called a virulent phage. Okay, a virulent phage is one that's going to kill the susceptible cell. So what happens is you infect the cell by means of interaction between the phage particle itself, produce a burst of phage as the initial cell lyses and dies, and that leads to infection of nearby bacteria, which can be visualized later by the formation of what are called plaques on a lawn of E. coli cell. So this is the lytic form of the cell cycle. Okay, so we can represent that fairly easily by having a cell. A phage attaches to the surface of it. It then inserts the DNA into the cell, leaving the phage head behind on the outside. And then you have a very carefully programmed series of gene expression changes where you have synthesis of proteins, replication of viral DNA, synthesis of late proteins, and assembly inside the cell of the new virus particles, followed by lysis and then release of the new particles into the medium, which can then go and infect new cells. So what this looks like on a plate, or schematically really, if we have a infected cell that got one little bit of DNA in it will lyse that cell, proliferate inside the cell, creating new phages. That cell lyses open, and the new phages spread to the nearby cells, thereby initiating new rounds of infection. Each of those new cells becomes infected, lyses, and dies, and that infection continues to spread. So what that ends up looking like, if you consider a plate full of E. coli cells, if the entire plate was spread with E. coli cells, some of which were infected by the virus, and you incubate it overnight, what you see instead of colonies of formed E. coli cells is little holes in the so-called lawn of E. coli where the phage have then proliferated. This is a little bit clearer picture here. These are called plaques, which are clear spots where the E. coli actually are all over the surface of the plate, and the little holes in the so-called lawn are where the phages are. So you could stick a little pipette in there and grab it and get some phages out of there, because that's where the phages are, and they haven't spread to encompass all the cells on the plate. But those are called plaques. Okay, so in terms of transduction, we want to consider the phage called P1. It has a lysogenic life cycle, but it also has a lytic cycle. So it can actually exist in the cell as a plasmid for many generations, but it normally will undergo the lytic type of growth, which we just described, which will lead to or can lead to generalized transduction. Okay, the generalized transduction idea is that phage P1 inserts its DNA into the cell, leading to replication, production of new phage particles, assembly of new phage particles inside the infected cell, and then lysis of the infected cell, which can then go ahead and infect new cells. Now, as part of this process, the partially assembled phages need to recognize and package phage DNA into the immature phage particles to form these mature phage particles. And in P1, the way it actually inserts that, it can actually occasionally, by accident, take in a piece of bacterial DNA instead of phage DNA. And that's the basis of the transduction. If you consider this cell, which has been infected and now is assembling new phage particles, you have phage DNA in there for sure. It can be packaged into new phage heads. But you also have kind of broken up chromosomal DNA, which is also available for being packaged into the new phage. So most phages don't do this, but P1 does. With some frequency, it makes a mistake and packages a piece of bacterial DNA into a new phage particle instead of its own P1 DNA. So a phage particle like that is called a transducing phage, a transducing phage particle, which contains genes. Randomly, any particular piece of E. coli DNA could be there that can then carry those E. coli genes or bacterial genes to another cell. Now that's generalized transduction because any old piece of DNA can be carried by the phage. And it's also a feature of this kind of transduction that the transducing phage contains bacterial DNA, not P1 DNA. It's not a mixture. It's only bacterial DNA in the case of those transducing phage. And those make up less than 1% of the whole population of phage. So most of the time, the phage gets it right, but a small fraction of the time, less than 1%, it gets it wrong and puts a bacterial piece of DNA in instead. Okay, so this is probably most of what I just said. The amount of DNA that's packaged in the transducing particle, which is done by what's called a head-full mechanism. It packages DNA until there's no more going to fit into the phage head. It's about the same length as the phage DNA would have been, which is about 100 kb or so. And when the phage particle is carrying this bacterial DNA, it will transfer it to a new bacterial cell. Remember that a phage particle is just a little molecular machine which is designed to take DNA and transfer it from one cell to another. It's basically a little gene delivery machine, and the particle will do that regardless of what DNA is present inside it. The particle itself doesn't know that it's bacterial DNA, not phage DNA, or human DNA, or whatever DNA. It will just insert whatever DNA is in there into a recipient cell. And in the case of P1, it's only bacterial DNA present in there, no phage DNA. So there's no possibility of the phage or the host cell breaking open or getting infected because what it's getting is not phage DNA, it's bacterial. Okay. Here's a picture. More than 99% of the phage are actual phage, where the phage has packaged its DNA into new particles, assembling them inside the cell, but that at some frequency you get, instead of phage DNA, bacterial DNA. And that will then be transferred to another cell. And it's generalized because any piece of DNA can be carried. So what happens when this transducing phage meets with a bacterial cell is it will bind to the bacterial cell by means of its tail proteins, etc. And then it will inject the new DNA into the recipient cell, in which case it's bacterial DNA. Well, to recognize and see this sort of event, again, we have to have recombination between the bacterial DNA and the chromosome. And in order to observe this, we have to set up some kind of a selection so we can recognize when those events have happened. So in this case, we have an A- recipient of some sort. So that represents the bacterial chromosome. A- referring to something like a Lu- or Trp- or something cell. And A+ meaning that the donor DNA from the now deceased bacterial cell that was infected by the P1 can then come in, recombine with the chromosome, and use the A+ to replace the A- that was there in the first place. And that's something that we can recognize or observe by setting up the appropriate selection with the appropriate genetic markers. Okay, so the idea is that you then transfer this A+ piece of DNA into the recipient chromosome, and then the A- that was originally there, say it was a Lu+, now it's Lu-, now it's a Lu+, that will be degraded, and that will then become part of the recipient genome. So that's something we can select by taking an initially Lu- cell and leucine auxotroph and plating the cells on medium lacking leucine and asking them to be able to grow by virtue of having acquired this Lu+ from the original donor cell that was infected by the P1 phage. So this is done by homologous recombination with the chromosome. It's just exactly like recombination with donor DNA from transformation or from donor DNA introduced by HFR. It's the same process catalyzed by cell proteins, which normally are involved in homologous recombination and repair. And this process does not require phage proteins, except for the fact that you need the phage head and tail to deliver this DNA into the recipient cell. And again, this is generalized transduction because any bacterial gene can be transferred. Okay, again, the selection is the same idea. We have to be able to select or identify what we can use to complement the auxotropies with the donor DNA, but then provide the same, whatever the requirements are for the cell, to grow anyway, despite the one thing we're selecting for. A transducing particle of a generalized transducing phage, such as P1, is composed of protein and phage DNA only, protein and bacterial DNA only, protein phage DNA and bacterial DNA, protein only, DNA only. And of course, it's B. It's protein, which is encoded by the virus, and bacterial DNA, which is being carried along to be transduced into a new recipient cell. Transduction is different from the other modes of gene transfer because only transduction uses a phage, involves DNA from the environment, requires cell-to-cell contact, requires cell surface pili. Of course, utilizes a phage. All right, this is a little more into it. Phage P1 was grown on a prototrophic E. coli strain that is resistant to the drug kanamycin. The phage were then used to infect an Rh-, Ura-, Lac-, kanamycin-sensitive strain of E. coli. What media would be used to select kanamycin-resistant transductants? Okay, I would suggest for that a rich medium with kanamycin so that the auxotropies don't make a problem. It would be rich medium with glucose carbon source along with kanamycin. Lac plus transductants, we want to select for the ability to use lactose as a carbon source. So we would actually have to include a minimal, well, it could be rich, let's say rich medium with the lactose as a carbon source, no glucose. Now remember that Lac plus means you can use lactose. A Lac minus cell cannot grow on lactose as its own carbon source. So if you plate those on lactose media, lactose plus media, no glucose, only lactose plus are able to grow. Or you could set it up with minimal, supplementing the auxotropies as you go. Rh plus transductants, you would need a minimal medium, no arginine, but it would include uracil. So it would be minimal plus uracil plus glucose. No kanamycin either. So you just need to look at the genotype of the donor, genotype of the recipient, and what it is you're trying to identify, and just set it up appropriately. I think that's probably what I just said. Yeah. Which of the following packages DNA into a protein particle by a Hedville mechanism? Phage P1, phage lambda, F factor. P1, okay.